এই সামনে পড়ে যাবে, ওই দিক দিয়ে ঘুরে আয়, এই দিক দিয়ে দাঁড়া।
আরে কাট করে সেন্টারে তো হচ্ছে না।
তোর এই ক্লিপটা খারাপ আছে, ঠিক আছে, প্রবলেম হচ্ছে কিছু?
এ ভাই কেমন একটা ট্যাড়া লাগছে।
বুঝতে পারছি।
আরে এইটা
এখানে ক্লিপটার জন্য প্রবলেম হচ্ছে। হচ্ছে না এবার ঠিক আছে,
ঠিক আছে।
ছেড়ে দেন, ঠিক আছে, বুঝে এখানে লাগছে, এখানে ঠিক লাগছে।
শুধু একটা ভিউ আসে তুই
আমি
নাকি?
সেটা
লাইট আইনি ভিউ আছে। আমাকে এটা পাঠালেন, সামনে চলে আয়। এমনিতেই শুরু হচ্ছে দেরি করে। আমার তো ফোনে কিছু দেখাচ্ছেই না। হ্যালো, ভালো কিছু হয়নি। তোর হাতে আমার মনে আমার ইউজ করিস না।
ঠিক আছে।
হ্যাঁ এবার হচ্ছে দুটো ভিউ আর নাকি? হ্যাঁ কোন ইয়ার?
প্রবলেম থাকলে বেরিয়ে
ভালো
কিছু না থাকে আচ্ছা না মেয়ে যখন ইচ্ছে না অন্য থাকে আমাদের বীজটা ছিল তখন 5টা 15 ট্রেন ধরবো ঠিক আছে না দরকার নিয়ে গেল
হচ্ছে
ডিলে করা আছে
তোরা ঢুকে একটু লাইক ফাইক দিয়ে দে।
আমারই লাইক
তুই কি করছিস? হ্যাঁ
আমি আর
ইউটিউব এনেবল করে রাখেন
এইটা কার চ্যানেল ভাই দাদা?
ডিপার্টমেন্টের চ্যানেল
তো তোরা ইয়ে করেনি কেন এটাকে?
তারা
সাবস্ক্রাইব করেনি কেন?
জানি
আর
না
কালকেই জানতে পারলাম এটাও আছে।
জানলাম যে ইউটিউবে
কালকেই জানতে পারলাম এরকম আছে।
বলে দে না, চারিদিকে ছড়িয়ে দে, এই ছড়িয়ে দে খবর।
আচ্ছা ছড়িয়ে দেবো।
এগারোটা বাজলো?
আমি বলে করে দিই ডিলে করা আছে ডিলে তোর 3 মিনিটের মতো ডিলে করা আছে
বাহ সুন্দর দেখা যাচ্ছে। কত দেখছি একজন? কত?
রাধা কি মেল করেছে
এই সেকেন্ড ইয়ার এগুলোকেও জয়েন করতে বল না,
হ্যাঁ
অন্তত জয়েন করে থাকুক।
জিনিস থেকে হচ্ছে পরের বছর থেকে বাড়বে সাবজেক্ট আছে একটা সাবজেক্ট প্রেজেন্টেশন 100 মার্কস
এই ডিপার্টমেন্টের যে ইউটিউবের চ্যানেলটা, ওটার লিঙ্কটা আমাকে হোয়াটসঅ্যাপে পাঠাবো।
ওরা তো পার্টিসিপেট করি মেয়ার পয়েন্ট কোনো গ্রুপে আছে
আমার গ্রুপ তুমি দেখতে
তাহলে তো পয়েন্ট করো
পারবো না
না সিআরপি জিজ্ঞেস করেছি
Good morning to all the eminent dignitaries, the esteemed faculty members, and all the students present out here. It gives me immense pleasure to welcome each and every one of you from the Department of Biotechnology, Haldia Institute of Technology, to this national seminar on New Horizons in Biotechnology 2023. This is a momentous occasion where distinguished experts and scholars have come together to exchange ideas and insights on various aspects. Today, we feel honored to have with us our Honorable Dean Ma'am, Professor Radha Das, School of Food, Chemical and Biotechnology of Haldia Institute of Technology. Our eminent head of the department, Professor Shubhrama Gupta, Head Department of Biotechnology, Haldia Institute of Technology, all the esteemed faculty members and students. This session will be chaired by our Dean Ma'am and our Head of the Department, Shubhrama Ma'am, will be co-chairing this session. I would request both the professors to please come up on stage.
একটু লেটে চলছে চ্যানেল।
আমি জানি ডিলে করা আছে
In the past century, the pace of technological progression has reached staggering rates to an extent that the field of biotechnology is constantly being disrupted with new innovations, developments, and breakthroughs. This seminar will provide a unique platform to explore and discuss the current trends, challenges, and opportunities in various interesting fields. It is an opportunity to learn from the experience, expertise, and perspectives of the achieve great understanding of the topic and develop innovative and practical solutions to the challenges faced in this area. Now, I would request our first notable speaker, Mr. Anupam Ash, Senior Quantity Control Manager at Indian Oil Corporation Limited, who has more than 25 years of experience in this industry. All of us by showing that academic pursuit is not constrained by age limit. Mr. Ash has repeatedly demonstrated his ability to strike a delicate balance between his professional and cultural endowments. Today, he will be providing all of us a basic review of the petroleum sector, its difficulties, and the best course of action for overcoming them via biotechnology. Now, I would request Sir, please come up on the stage.
কথা তো আমাদের এগিয়ে আছে কিছুই কথা নেই তো
Good morning
Mr. Strong আছে
to this August gathering. I am in an academic institution after more than 30 years. In front of us, the reputed professors, my respected teachers, and my beloved students.
এগিয়ে আছে দিলে আছে
Thank you for calling me to be a speaker in this New Horizons in Biotechnology Seminar 2023. I really feel proud. Being a technical background with the machineries, we generally talk about the machines, this is not working, why this is not working, why the production is shut down, why production is not dispatched. But here, the thing is something different. We are here to change the world. Change the world with an idea, a simple idea, the out-of-box thinking. We all do the in-box thinking, but you, the people here, those who think out-of-box, I really salute this attitude. Today's seminar is on new horizons in biotechnology. I am not in the field of biotechnology. I am a man of little bit knowledge of organic chemistry and petroleum industry. So, I will provide you a general overview on the petroleum refining industry first, so that after my lecture or after this seminar, you will accumulate some information about the refining processes and what is going on in the refinery. Refinery provides the energy to the wheels for the progress of the nation, for the progress of the mankind, for the better civilization. I also thank to the great personality whom I have seen in the year of 1993 when I came here at Haldia, Dr. Lakhan Singh, for whom only this institute is now standing as an iconic institution in West Bengal in particular and in India at large. I hope you, my beloved students, you will carry the legacy of this institution. Haldia Refinery is the fourth refinery of Indian Oil Corporation Limited in Chennai. We have 10 total refineries. It is the first East Coast refinery to mitigate the oil demands of the eastern region, particularly West Bengal, Bihar, part of Bihar now it is Jharkhand, Odisha, Sikkim, Nepal and Bhutan. It was started in the year of 1975, though the foundation stone was laid down in 1969 by the then Petroleum Minister Dr. Triguna Sen. 5-6 years it took time to build up. The main aim was to provide the lube oils to the mechanical industry in India. The only lube oil manufacturing refinery in India to provide the lube oils to the machinery, because for the industrial revolution in the 60s, 70s, the huge crisis of lube oil was there. In refinery, the starting material is crude oil, petroleum crude oil. But in India, the resource of crude oil is very less. It is almost 2% of the need. And that 2% is mainly used in the Assam region by the Assam Oil Division at Decoy, mainly, and few portion is used in the Guwahati refinery, few portion in Numaligarh refinery, few portion in Bongaigaon refinery. That all are in Assam. So we import all the crude oils. As it is a business sector, I will provide you some figures. But because I am from a business sector, the total crude price is the 90% of the sale price. For Indian Oil Corporation Limited, we need 80 million metric ton per annum crude oil to process, which cost at present today 8,50,000 crore. And total business volume is 9,44,444 crore. 2% is the operational cost. 3% is the other miscellaneous cost and loss, and 5% is the profit. 5% means almost 50,000 crore. It is the profit of the Indian Oil Corporation Limited in a year. That is the revenue generated in the fund of Indian Government of India. So Indian Oil Corporation Limited generates a huge purpose to the Indian economy. So this crude oil, 90% crude oil, came from mainly Middle East, Saudi Arabia. Our foreign relation is very good with Saudi Arabia. Middle East, Kuwait, few portion from Nigeria, and few portion from Russia. Russia crude is cheap. All this crude came through the VLCC, very large closed container, to Paradip. And from Paradip, there is a crude pipeline, Paradip, Haldia, Barauni crude pipeline. Through this pipeline, material came to the refinery, and refinery will process it in our different processing units to get first LPG, the requirement in the kitchen. Then all your petrochemical industry, which their base stock is naphtha. All the plastic goods which we are using in our daily life, the starting component is naphtha. Then the kerosene. Kerosene we are not using in our day-to-day life. All the kerosene is now converted into aviation turbine fuel. ATF required for the flights, and definitely for the Indian Airlines, for the fighter planes. Next is the diesel, and of course it is petrol. Petrol and diesel is the main fuel for the automobiles. Next, now it is not used in too much, that is jute oil. Once upon a time, in the two sides of Ganges, there was a jute industry and huge demand. There was huge demand of jute batching oil. But now jute batching oil demand is slashed down to only 10%. The jute batching oil is also a product of Indian Oil Corporation, particularly in Haldia refinery. Next is furnace oil. The ship is moving, marine line, coastal line. The fuel is furnace oil. That furnace oil we produced. There are mainly three ways of transport. One is road transport. For road transport, we use petrol and diesel. For air transport, we use aviation turbine fuel, and for marine transport, we use furnace oil. All these products are being produced in Haldia refinery in particular, and all refineries in Indian Oil Corporation around the nation. Now, the question is, we all know this reserve of crude oil is depleting day by day. And for this crude oil, globally there is a turmoil in political hitch in between the muscle countries. America fighting with Russia, fighting with not your Arabian countries only because of this oil. But this oil reserve is going down day by day. And we know the carbon footprint we have to reduce as to reduce the global warming. So alternative energy is required to protect the environment. We also continue to make big strides towards reducing the environmental footprint. Our approach towards environmental stewardship encompasses measures and initiatives around emissions reduction, increased water and energy conservation, waste reduction, and increased usage of renewable energy. We are also investing towards building presence in the green energy segment that empowers our customers to reduce their environmental footprint, especially the green fuel, electric vehicle, battery, and hydrogen. To manage the environment, we are reducing the fresh water consumption and we are trying for the zero discharge water to the river or sea. Here, as it is a biotech seminar, to treat the wastewater, there is a huge role of the biotechnologists. Now, in Indian Oil Corporation Limited, there is a research and development section in Delhi, Faridabad. They are working to produce different type of bacteria which will grasp the oil or sulfides which are present in the wastewater to reduce the carbon, your chemical oxygen demand, biological oxygen demand in the wastewater. So that research is going on. So it is a wing for the biotechnologies to support the refining section to reduce the environmental impact. As a public sector enterprise, we have the duty to protect the environment. Already 20 lakhs mangrove planted in the delta of Hooghly River opposite to Haldia Refinery under corporate social responsibility project. Dense afforestation following Miyawaki concept. It is a concept for deep afforestation at a very minimum time. The sample is in the Mohisadal Rajbari area. You can visit Hooghly River. Collaboration with the Department of Forest, Haldia Development Authority and Haldia Municipality planned a cumulative 60% green coverage by 22-23. Alternative energy. Alternative energy, Indian Oil is also working on alternative energy, and the government of India is also suggesting for the blending of biodiesel in the diesel. 10% is allowed. So it is also another wing for the biotechnologists to mitigate this 10% demand. That is also huge biodiesel. That is from Jatropha, this type of thing. We are getting waste product. This is our waste product, and we process it in the Tetulbedia area in HPL Link Road, and we mix with the diesel 10%. That is biodiesel. We also blending as per the guideline of the government of India 10% ethanol with the petrol, and just few days back government is suggesting please try to add 20% of ethanol and see what is the changes in the quality of the petrol. We are working on it. We are adding 12%, 15%, and 20% haze-wise, and we will see what is the change in the fuel quality. Oh, this ethanol will be produced in the Paradip refinery, very close to Delhi, only 90 kilometers from Delhi, in the Haryana region towards Kurukshetra. We are producing, we'll start producing by 2023 from the paddy hay and your cane sugar waste to produce ethanol. Indian Oil is spearheading the goals SATAT scheme. SATAT scheme is sustainable alternative towards affordable transformation. It is the scheme of the Government of India with the target of 15 million metric ton per annum CBG production. CBG is compressed biogas. Indian Oil is also investing in the field of wind energy, in the field of EV to produce the battery. Indian Oil has resolved to achieve net zero operational emissions by the year 2046, before the completion of 100 years of independence. 100 year of independence in 2047, we have the target by 2046. The carbon footprint will be zero to the environment. There will be no flare. We usually see a flare in the refinery. That flare will not be there. Flare is the emission. So we use the flaring components to convert it into usable components so that there will be no emission to the environment, that is called net zero. The year 2046 coincides with India completing 9 years of independence and the celebration of its 100th Independence Day. The historic declaration by the country's energy major aligns with India's net zero commitment by Prime Minister in the year 2070. Prime Minister declared it in the year of 2070, the emission will be zero. But Indian Oil taken the oath, will do it by 2046. As a part of the Panchamrit goals by the Honorable Prime Minister at the 26th UN Climate Change Conference Summit last year. The 7 focus areas of Indian Oil Corporation: achieve net zero operational emissions, meeting electricity requirements of expansion projects through renewable energy, increase the proportion of cleaner fuels in the product mix, invest in low carbon energy technology and undertake partnerships, engagement with value chain partners to reduce emissions across the current network, bring greater linkages between employee skills and company climate goals, establish Indian Oil as a leader in environment sustainability. These are the 7 focus areas of Indian Oil Corporation Limited. As it is a biotechnology seminar, and I will try to link up the biotechnology with the refinery, where the biotechnologists can play a pivotal role to help the refineries. The petroleum industry plays a crucial role in our economy and society, providing the energy needed to power our homes, businesses, and transportation systems. However, the industry also faces significant environmental and economic challenges, including the depletion of fossil fuel reserves, environmental pollution, and climate change. Biotechnology offers a promising solution to these challenges by providing sustainable and cost-effective solutions to the petroleum industry. Biotechnology involves the use of living organisms and biological processes to develop new products and processes. In the petroleum industry, biotechnology is microbial enhanced oil recovery, biofuels, bioplastics, and carbon capture and utilization. Bioremediation is the use of microorganisms to break down the degrade pollutants in the environment. In the petroleum industry, bioremediation is used to clean up oil spills and contaminated soil and groundwater. Bioremediation is a cost-effective and environmental friendly alternative to traditional cleanup methods, which often involve excavation and disposal of contaminated materials. MEOR, that is microbial enhanced oil recovery, is a process that uses microorganisms to enhance oil recovery from reservoirs. Microorganisms can be injected into fluid and easier to extract. MEOR can be used to increase the efficiency of oil extraction and reduce the environmental impact of oil production. Biofuels are another area where biotechnology is making a significant impact in the petroleum industry. Biotechnology is being used to develop biofuels from renewable sources such as algae, cod, and sugarcane. Biofuels have the potential to reduce dependence on fossil fuels and lower greenhouse gas emissions. Biotechnology is also being used to improve the efficiency and cost effectiveness of biofuel production. Bioplastics are biodegradable that can be produced from renewable resources such as corn, sugarcane, and cellulose. Bioplastics have the potential to reduce dependence on petroleum-based plastics, which are non-biodegradable and contribute to environmental pollution. Finally, biotechnology is being used to capture and utilize carbon dioxide emissions from industrial processes. Microorganisms can be used to convert carbon dioxide into biofuels, bioplastics, and other useful products. This technology, known as microbial carbon capture and utilization, has the potential to reduce greenhouse gas emissions and mitigate climate change. In conclusion, biotechnology is a promising field that offers sustainable and cost-effective solutions to the petroleum industry. By harnessing the power of biological systems, we can develop new products and processes that address the environmental and economic challenges facing the petroleum industry. Let us continue to support and invest in biotechnology research and development to create a more sustainable future for all. Thank you.
Thank you, sir, for your informative talk. Now I would request Dr. Mukesh Singh, Associate Professor, Department of Biotechnology, Haldia Institute of Technology, to honor our eminent speaker with a token of appreciation. Thank you, sir. So, sir has given us an insight about how biotechnology is useful in the petroleum industry. So, does anyone have any question to sir? Any questions from the audience? Okay, well, thank you.
Our next esteemed speaker is Dr. Atin Kumar Manto. We would like to use this opportunity to speak a bit about his academic background and multiple achievements. Since 2020, Professor Atin Kumar Mondal has held the position of Professor in the Post Institute's Division of Biotechnology. Prior to it, he served as an Associate Professor in the same department. He worked as a Research Associate at the City College of New York's Biology Department from 2000 to 2010. He also worked as a Research Associate at the Worcester Polytechnic Institute, Massachusetts Department of Chemistry and Biochemistry from the years 2003 to 2005. His areas of interest for research include understanding the mechanism of protein kinase quality control in development with pathological conditions such as cancer and cardiac hypertrophy, crosstalk between molecular chaperones and ubiquitin ligases in quality control of misfolded aggregated proteins, role of Praja1 ubiquitin ligase in maintaining cellular proteases. He has his articles published in a number of national and international journals of esteemed reputation, such as PLOS One, Journal of Biological Chemistry, Journal of Cell Biology, and several more. Today, Professor Mondal will be giving us an insight about the role of molecular chaperones in proteostasis. I would now request Sir to come up on stage.
Good morning, everyone. So, First, I'd like to thanks to the organizing committee of New Horizon of Biotechnology 2023 for inviting me here. So today, I mainly emphasize how a efficient quality control machinery that usually we have in the industry to see whether the product, actually finished product, is really good for marketing, also exists inside the shell for our surveillance. So basically, how a proteostasis. So what does it mean? It actually maintains the protein that synthesized or degrade all the time inside the shell is maintained for keeping the homeostasis in balance. So what does it mean? It's just essentially a process that maintains protein homeostasis inside the shell. So people think, as we know, the proteins actually synthesized at the ribosome. So when a protein synthesized with the ribosome that we call newly synthesized proteins or nascent polypeptides, it usually folds to its native conformation. And the native proteins or folded proteins has specific in their respective place. But 3 to 5% of the newly synthesized proteins always at risk of misfolded. And those misfolded proteins also can form if you have a stress condition, any kind of stress, protein gets misfolded. So that have the proteins as misfolded, and these misfolded proteins are always risk to having aggregation. And if you have aggregate, protein is aggregated inside the cell, that can cause several proteinopathies like several neurodegenerative diseases what we have right now, like Alzheimer's disease, Parkinson's disease, Huntington disease, and all these things. So those are mainly misfolding of the proteins that we cannot clear. Not only that, so shell has an efficient mechanism to clear those misfolded proteins. And what are those? The shell actually tag those proteins, recognize those misfolded proteins, and tag with a system called ubiquitin. With the help of ubiquitin ligase, specifically tag misfolded protein with ubiquitin and put them subtly to the degradation machinery. What we have like proteasomes, ubiquitin proteasome system, or it can be engulfed with the autophagic lysosomal degradation. So these two systems, chaperones and degradation machinery, keeps the balance proteome or keep the proteome in balance. But if any kind of stress happens, like for example in genetic mutations, that usually we have often in aging, because all the quality control system actually keeping in low. So you have frequency of having genetic mutations or oxidative stress or any kind of environmental stress. It can be heat stress, it can be toxicity with any metals that causes proteostatic stress. And it disbalances the protein homeostasis. And as a result, we have proteostatic stress and the protein machinery, like control machinery, doesn't work anymore. So we have accumulated of misfolded protein inside the cell, and that actually helps for disease progression. So, as we know, probably, the protein actually folds by itself, right? This is a classic experiment with Christian and Finchens. What he did, he took a protein, urea or any catatrophic agent, and protein can misfold. But if you withdraw the degenerate agent, it can refold and protein can start working again. So protein actually can fold, so this suggests that it's a self-assembly of the proteins and which is continuous. Okay? However, the principle of self-assembly of proteins that requires optimal conditions in vivo for favorable cellular environment. However, if we think in a cellular perspectives, for example, if you take the E. coli cells, It's a tiny volume, 0.2 to 2 microns or 3 microns. And how many proteins it has? It has over 4,000 proteins with this tiny volumes. If we go evolutionary to yeast, like Saccharomyces cerevisiae, like the fake and it has over 6,000 proteins. And in man, we have 20 to 30 proteins. And the average protein length also increased from E. coli to mammals. So with this tiny volume, we have enormous number of proteins. And not only that, so there is a molecular crowding. And also, we have relatively little bit high temperatures that actually unfavorable for protein folding inside the shell. So, protein cannot fold by itself within the cell, which we see in so here it comes the molecular chaperones, which is called assisting proteins, and what it does, it helps facilitates folding of new synthesized proteins and helps not only it acts as a key player to keep the proteome in balance. So it helps newly synthesized protein to fold. Not only that, it maintains the functional conformations of the proteins that can form proteins to be different kind of aggregates. In addition, chaperone also with the other proteostasis network, it can clear the misfolded proteins via autophagic system or the proteinase systems. So all these functions is actually maintained by the molecular chaperones. And if we think it's the protein folding landscape, how chaperone works here? So this is the protein folding landscape. It's a folding funnel, and the energy is increasing on the folding. So initially, when protein is synthesized, we have several number of unfolded conformers, and now we have only single netic conformation that is stabilized. So initially, you have multiple numbers of conformation, and with the folding funnel, which is not smooth as we see in a funnel, like any kind of funnel is smooth, but in protein landscape, it's not smooth, it's a round surface. And you have energy minimum in any cases. And holding intermediates can be trapped anywhere. Can be trapped anywhere. And what chaperone does here? Chaperones actually lowers the energy barrier. So it helps to facilitate to attain this native state. And also, it prevents any kind of folding intermediates that can be trapped inside the interfolding off pathway. So thereby, it helps in the folding process. So why we need to study this? Because it helps to keep quality of the proteostasis or maintains the proteostatic quality, which is usually decreasing aging or in any kind of diseases. It promotes protein folding, which is essential, which when protein misfolds, we have in aging conditions, we have several developments of several neurological diseases. It also acts as a sensor, which if you have a misfolded proteins, then it cannot sense the stress. So we have a diabetes, what we have right now in most of the families. Also, it acts as a buffer. In actually keeping the morphological traits or the phenotype from one generation to other generations. I mean, it also works to stabilize the inheritance. Okay, so what these are molecular chaperones? So these are a group of proteins. Those are highly diverse, and they actually bind to the hydrophobic stage of the proteins. So as you know, the hydrophobic amino acids, those wants to stay inside of the protein because it can interact with any other stage. So it binds to the hydrophobic stage and binds to unfolded proteins in order to facilitate protein folding and also prevent non-specific interactions. Not only that, it unfolds the protein during translocation to the one compartment to the other compartment, like when protein translocates from in any component like ER, mitochondria, chaperones actually helps to keep the protein unfolded for the transport in different organisms. So this happens first described as a heat shock proteins. If you have a heat shock, chaperones actually go off because you have lots of protein gets misfolded, chaperones that opens up the hydrophobic stress as chaperones wants to go and bind there. So cell actually keeps chaperone level up during stress conditions. But later on it was categorized with their molecular mass. So they have various families like small hits of protein with diverse family from 10 to 30 kilodaltons, what we have like 26 or 27 like alpha dystrolane what we have in our eye. Then you have different molecular chaperones, HSP 40, 60, 70, 90, and HSP 100. So those are the different family of molecular chaperones exist in cytoshells. And these chaperones are evolutionarily conserved from bacteria, yeast, and mammalian systems. And these chaperones, when newly synthesized proteins come inside of the ribosomes, immediately chaperone works at the ribosomes. It binds to the newly synthesized proteins in bacteria called trigger factor, in yeast, same as RAC and MAC complex, RAC complex or nascent chain activated complex, same as in mammalian system. Then, after ribosomes, cellular cytosolic chaperone system comes into play, which are HSP70, HSP40, in case of DNA K, DNA J in the bacteria, in mammals, HSP70 and 40, and same thing for mammals. However, in bacteria, it also acts as GROEL system. In yeast or mammals, it was called TIC60 system. So it's the same from bacteria, yeast, and mammals. So basically, newly synthesized proteins recognized initially by HSP40, then proteins is transferred to the HSP70. Some proteins can fold with these two chaperone systems. Some proteins require further chaperoning with the help of GROEL system we call chaperoning, that is a dumbbell cell, where you have a chamber, proteins can, unfolded proteins can get in, then it can fold to the native state. Some proteins require HSP90 systems, and those have specifically for different substrates. For example, for several transcription factors, protein kinase, and all these are highly are folded by the HSP90 subpositionaries. So how this cycle works? So misfolded proteins or unfolded proteins, when they are synthesized from the ribosomes, so chaperones actually binds and release, and this is a kind of cycle. Initially binds to the 40-70 called early complex. Then we have another protein called HSP organizing protein that recruits early complex to the intermediate complex that recruits HSP90. Then 70 dissociates, and we have a late complex, and the further proteins can be folded. And how we can see it in shell. So first description of this cycle was given. So this is kind of experiments, how we can see it. So this is a checkpoint kinase. It's a protein kinase that works in the initial cycle. And what they did, they purified the proteins with a glutathione. So if you put a GST or glutathione phosphorus tag with the protein, so you can bind with the bead and purify it. And binding of the several chaperones. So what here they are seeing binding of molecular chaperones in a time dependent manner. What we see initially 70 binds, then it's decreasing. Then another chaperone CTC37 comes in, started to bind and then increase. Then 90 it comes again later and highly at the end. Do it in functional activity, when 90 is binds, activity is high. So this is kind of initially 70 binds, then these two players comes in the play for this. So what we are trying to look at it, how this happens actually play in holding up protein kinase, and we are looking at it in yeast species, because yeast has almost 123 protein kinase in various families. And we are going to look at how this happens actually helps in their folding. So initially we looked at the HSP90, which is a master chaperone, master regulator. And if we want to inhibit these chaperones, there is a drug called clindamycin, and it binds HSP90 binding pocket. And what it does, it cannot hydrolyze ATP, so protein cannot fold. Then if we treat the yeast cells with this drug, what happens? And we are looking at the kinase that is tagged, and also we are looking at the newly synthesized kinase. How? We label the kinase with radioactive. Then we are looking with the immunoprecipitation with status of the kinase with time dependent manner from 0 to 2 hours. What is the level of the protein? And if there is no drug, protein is stable. If we inhibit, apply the drug, we cannot see the protein after half an hour. After this goes. So protein actually called degrade. If we block the proteasome with a drug called angioam32, we can rescue the protein. That means protein, if you inhibit HSP90 functions with this drug, it cannot go to the folded function. It's actually degraded. So next we want to look at what is the role of this CDC37, which actually acts as a recruiter. So this CDC37, it's a eukaryotic molecular chaperone, is highly essential. If you knock down it, cell will die. So, and it has two functions. One, it can bind to the substrate, and also it can bind to the HSP90, which is the domain. And it acts as a HSP90. And interestingly, these chaperones actually upregulate in various kinds of cancer cells or tumor cells. If you wanted to look at it in a global manner, how this chaperone works. So what we did, we delete endogenous CDC37 from yeast. And if you look at the mutated one, version, introduce the mutated version in the genome, which cannot bind to the kinase. And then we looked at it, individual kinase status by a Western blot. We are just looking at the level of the kinase, introducing a mutated version. And we checked 65 kinases. And out of that, almost 51 kinases we see has reduced level. But some, there is no change. So there is a global, 75% kinase actually dependent on CDC37. So if you remove CDC37, the proteins actually goes for the degradations of the proteasomes. However, some kinase doesn't need CDC37. So next we want to check what happens to the other chaperones in the folding pathway. And we are looking at the early complex, where is the HSP7040 or others. And we also check another kinase, status of the kinase by knocking it out. Endogenously, you can knock down or delete the chaperones in yeast very easily. Like what we do in the mammalian cell like CRISPR or SAEs or CRISPR you can knock out or SAEs or sequencers. So maybe if we had knocked it out and we see one chaperones like YDG1 which is HSP40 and we have reduced level of the kinase. So what do you want to see? What happens if we knock down several kinases and check the level of the kinase in this knockout state? And we see several kinase level is actually decreased in HSP40 knockout state, and which at the same that is dependent on CDC37. Some kinase that doesn't require YDG1, it also doesn't require CDC37. And how to check it, whether it is degraded or not, we did level it and check its level with time dependent manner. in wildlife and knockout cells, and what we see in 2 hours actually proteins is degraded in a knockout cell. So this YTJ1 or HSP40 in yeast actually requires for stabilization. That means it helps in the protein folding. So what we show is that YTJ1 or HSP40 or series has both is required for this folding in presence of HSP. And when you do not have it, proteins actually goes to the degradations. Now we want to check it. What is the role of HSP70 here? Because HSP70 actually comes in the beginning. If we inhibit HSP70 with a specific drug called myrisipine or fish, that drug is, we can find it is a polyphenol. So if we treat the yeast cell with this drug, we see there is a change in mobility. Increasing drug concentration, mobility is actually changed. And what it does, this kinase actually can have a autophosphorylation sites. If we mutated this autophosphorylation, you cannot have this mobility change. So, what it suggests that if you have the drug, it cannot actually mature. Not only that, if we apply the drug, kinase is not at all functional when you check the kinase activity. So, 70 inhibition actually prevents protein folding. Okay. So what we show for sure that is the protein folding or the kinase folding requires HSP40, 70, CDC37 and HSP90 for in the folding process. Now what happens if you have deep stress? To misfold the proteins. If you put the stress conditions like put heat stress, protein misfolds, and misfolded proteins can form aggregates, and here we tag a fluorescent tag called GFP with the kinase, which we can see in the fluorescent microscope or confocal microscope. And when it's in the normal temperature, where yeast grow like 30 degrees, but if you put a heat stress in 37 degrees, the proteins actually forms in aggregates, which we can see in the microscopes. And these aggregates also depends on the chaperones. In these cases, HSP42, because HSP42 also another chaperones that actually localize. It also can be aggregated with the heat stress. Similarly, if you have inhibit HSP90, protein degrades, but it can form aggregates. Same thing with HSP70, but this in stress conditions, these aggregates forms differently in these cells. They can be close to just a nuclear position. Those are highly ubiquitinated and can be degraded. It can be a stress foci or that is highly mobile, which are colocalized with 42, and that can be refolded or degraded. And there could be another aggregate, kind of aggregate, those are insoluble, highly insoluble and non-ubiquitinated. So what is the fate of this protein? If you have a stress, you have protein is sequestered, it can be refolds or it can be degraded. So when it is degraded to proteasome or autophagy, what the chaperones actually require for that? So we look at the HSP70 here. When you inhibit HSP70 with the same drug, we can see proteins actually stabilized. This is the pulse chase experiment. With time-dependent manner, you are looking the status of the proteins. With 6 hours, proteins actually disappear, but if you block HSP70, protein gets stabilized. Same thing, we did it in a knockout cell or temperature-sensitive cell in a genetically manner, protein gets stabilized. That means the HSP70 also required for degradation. It's not only required for folding, it also required for the degradation of protein. And how it does? It actually helps to ubiquitinate the proteins. If you have a mutated strain, the ubiquity will decrease. Same thing for high temperature. If you increase the temperature in presence of knockout strains or mutated strain, the level of ubiquitin is substantially increased. It suggests that 70 actually required for ubiquitination. formation of the misfolded protein for the degradation. So this 70 works alone because 70 it also has the ATP hydrolysis power that is mediated by these two proteins called HSP40 and HSP110, which is called SSC1 pair. So then we look at what is the role of SSC1 pair. If we knock out SSC1, then we misfold the proteins, in knockout cell protein gets stabilized. That means not only 70, these proteins or HSP110 class of proteins also require for this degradation. How we can see it? When in a knockout state, we are looking at the ubiquitin level, and here we put a drug called agitated carboxylic acid, that is a proline analog. If we put that drug in protein in polypeptide, instead of proline, it will incorporate agitated. How? And in that case, protein will be misfolded. If it is misfolded, then it can be ubiquitinated. So when you have applied AGC, you see ubiquitinated level is increasing. But in the knockout state, the ubiquitin level is decreased. Similarly, we see in a particular protein called cell level, in knockout state, ubiquitin level is decreased. So SSC1 also involved in the process, and that also requires HSP7. So what we have so far we have said, in the protein folding process, we have HSP40, series 37, and HSP90 actually involved for the folding of protein. However, if we needed HSP90 with the specific drug, proteins can be recognized by the HSP70 with the SSC1, it can transfer the position to the mechanism for the degradation. Now, what is the role of E3 ligase? What E3 ligase works? And we identified that two ubiquitin ligases called UBR1 and SHAN1 in yeast that helps, if you knock out it, it actually stabilizes the proteins for UBR1 and SHAN1 in different strains. So these two ubiquitin ligases involved in ubiquitination of the misfolded proteins. If we delete it, ubiquitin is substantially goes down. So in conclusion, what we show it, folding or kinase quality control is maintained by the chaperones, where several pool of chaperones actually required for the folding, and HSP70 works as a both phases. It requires for the folding as well as for the degradation of the system, and that is transfer the substrate to the E3 ligase for the degradations. Okay. So why it is important? Chaperone is important. Right now we are seeing that in several cancers, these chaperones are upregulated. Particularly HSP90, 70 series, and also called OP, which is the assisting proteins. All these chaperones are upregulated in cancer. Why? Because they want to stabilize the metastatic conformation of proteins. And all chaperones are currently in the drug target. They have some, like HSP90 has a phase 3 clinical trials, also currently like series H. Conversely, in neurodegenerative diseases, protein quality control is substantially down. Mainly HSP70 level is quite down. So now we are trying to see whether you can have a hit shock response that actually increase the level of the 70, which can be utilized for degradation to pull the protein misfolded protein degradation. So people are actually trying to work in there whether we can give the hit shock response or increase the HSP70 level in a sense to mitigate the misfolded protein pool that could be helpful in protecting neurodegeneration. And not only that, chaperone also helps in biotechnological applications because several proteins we have difficulty in purifications in a mass scale. So how chaperone does? Chaperone actually keep the protein in soluble conformations. That will be helpful in the large scale production of any kind of proteins that is helpful. So with that, I thank the I thank you I can stop here. If you have any questions, I'm happy to answer.
Thank you, sir, for an interesting and engaging lecture on molecular shuttles. Once again, I would ask the audience if they have any questions regarding this topic. Yeah, please say.
এই সঞ্চারী
Good afternoon, sir. My question is, how can stresses in protein cause neurological disorders? Proteins.
Hello. Sorry,
How stresses in proteins, stresses meaning proteins that are unable to fold, can cause neurological disorders?
Okay. Neurological disorders mainly happens, there are few proteins, like those causes neurodegenerative disorders like Alzheimer's disease, that's called responsible for the A beta. A beta actually can get misfolded in several cases. Like if you have oxidative stress, A beta can be misfolded and attains A beta 6 structures. Normally it is fine. It helps in normal cellular function. If you have any kind of mitochondrial stress, like oxidative stress, A beta can be misfolded and it attains a beta 6 structures. As a result, it forms the aggregates that is insoluble in nature and it cannot be degraded. So it attains as a misfolded conformations and it helps actually if proteins aggregates, then it changes the quality control, means mitochondrial dynamics and everything will change.
Thank you, sir.
Thank you for your question. Anyone else? Please.
যে কোশ্চেন আছে ও আচ্ছা
Hello, sir. Sir, whether this chaperone binds with nascent polypeptide in a sequence-specific manner or not?
No, it's usually the hydrophobic stretch. Like there are stretch of hydrophobic sequence. It only recognizes the hydrophobic stretch.
Okay, sir.
It's not the sequence list. Means there are some hydrophobic amino acids there. So 5, 6 or 10 amino acids where there is mostly hydrophobic, because hydrophobic states only can interact with the intermolecular interactions. So these states actually try to keep inside because it's a non-polar, right? So they try to hide it inside. So when protein synthesized, it is in the linear. So linear, if a stretch is linear, it can interact with other proteins very easily. So chaperone then protects, binds to the hydrophobic stretch and protects it from intermolecular interaction.
Thank you, sir.
Thank you for your question. Are there any more questions? Yes, please.
Trigger factor is the kind of protein that is exist in that signal. Its name is trigger factor. The protein name is trigger factor. So it's a cradle-like structure that when nucleosynthesis cells handle the ribosomes, it actually binds to the ribosomes and so it cannot interact with any other proteins. So it is like a cradle-like structure. Its called name is trigger factor in bacteria. Particular proteins, newly synthesized proteins. But it is absent in other means evolutionary in yeast and mammalian there is no trigger factor, but that is called RAC and NAC, ribosome associated protein complex or nascent cell activated complex. those are the two proteins, two group of proteins required for eukaryotes, not in prokaryotes. Prokaryotes is called trigger factor, but eukaryotes is RAT and NAC complex. The same kind of chaperones. Actually, what happens if you have any kind of changes, morphological changes or phenotypic change, chaperones actually keep the changes, try to keep the changes because you need to survive. So chaperone keep the and means what it does, it stabilizes the changes basically. Otherwise, let's say for cancer, let's say any kind of mutation happens during cancer, right? Chaperones actually helps holding those proteins. Why they are upregulated? Because they try to hold those proteins, and this protein is only dependent on the chaperones. So in that case, chaperones plays a buffer system to keep the homeostasis, and it also helps in a phenotypic change traits. If any kind of phenotypic traits evolve, chaperones also play a critical role there to keep the change. That is progressed in generation to generation. কৃষ্ণানন্দ ঠিক বললাম তো?
Thank you for your question. Any more questions? I guess not. So moving on, I request Dr. Kiya Shah ma'am, Associate Department of Biotechnology for the Institute of Technology, to felicitate our honorable dignitary with a small gesture of appreciation.
Thank you, Professor Atin Kumar Mandal. I have one small question. The thing is, you have said that for cancer, some chaperones, they can act as a target. You also have shown a drug, gentamicin, which targets those chaperones. My question is, in which type of cancer cell these chaperones can be acted as a target, and if those chaperones are used as a target, can they be used in combination therapy? Because we know that there are many different types of drugs attacking or targeting various proteins, molecules, tubulin, etc. There are so many DNA. If target, can those drugs can be used in other drugs also in order to increase the ICD demand?
Not only protein, there's lots of other problem there. Have only lipid 90. Drug targeting for a long time, years back, more than 2 years. It was identified in 1964. From there, it is shown and acted as a all the cancer, breast cancer, are two only BRAF mutation, melanoma, melanoma, G600 mutation, BRAF, I did clinical trial. Most likely they are trying to
সৌমদীপ তো সৌমদীপ সুনিকেতা ব্যাগ আছে ব্যাগটা দেখ সুনিকেতা
Thank you, sir. Moving on, our next esteemed speaker is Professor Dr. Bhashwati Pandit. I would like to take a few minutes to talk about her academic background and enumerate her various accomplishments in academia. Professor Bhashwati Pandit has done her bachelor's and master's in chemistry and biochemistry respectively from the University of Calcutta. She has obtained her PhD in biochemistry from the University of Calcutta. Her postdoctoral research at Medical University of South Carolina was in genetics of steroid metabolism and related disorders. She has also done postdoctoral work at Mount Sinai School of Medicine, New York, on the topic genetics of heart maldevelopment syndromes, namely the Noonan syndrome. Her work led to the discovery of RAF1 mutation, leading to hypertrophic cardiomyopathy phenotype in Noonan syndrome. Moving back to India, she joined the National Institute of Biomedical Genomics. Her research passion is to understand the genetic underpinning of disease, whether chronic, syndromic, or infectious. Besides congenital heart disease, currently her working area has also expanded to genetics of infectious disease, identifying host determinants of tuberculosis infection. Her work on tuberculosis has led to the identification of CXCL10, a chemokine as a marker for active disease. Her group has identified novel variants in SIG-LEC15, HLA-TRA associated with tuberculosis. This study was carried out in the population in and around Kollam. She is a member of several societies like the Indian Society for Human Genetics, Calcutta Consortium for Human Genetics, DNA Society of India, etc. She has several articles published in a number of esteemed national and international journals such as Mitochondrion, Infection, Genetics and Evolution, Tuberculosis, and many others. Today, Professor Bhashwati Pandit ma'am will be enlightening us about host genomic variations and its influence on susceptibility to tuberculosis. I would now request ma'am to come up on stage.
না এটা এখানে আজকে শেষ কালকে নতুন সব ডিফারেন্ট জায়গা থেকে কলকাতা জায়গা
Good afternoon to everyone, and I would like to this opportunity to talk about some aspects of the research work that we are conducting in our lab. So since morning, the series of talk, I feel that we are having like a chemical evolution. You know that there is a chemical evolution theory of life. So from biomolecules, you know Miller's experiment that at some point of time led to formation of macromolecules. So from petroleum, we heard about protein, and now I'll take you to a disease aspect. So particularly to the students, I would say the few terms in my title, if it is not clear to you, I'm ready to explain you, and I hope at the end of my talk, I'll be able to convince you. So, what is the disease tuberculosis? I hope all of you know, right? So, what is tuberculosis? Which pathogen has caused tuberculosis? Okay, all of you know. So, mycobacterium infection caused tuberculosis, and many of you know that the clinical symptoms are like somebody coughs, they have low-grade fever, weight loss, and so on. And for this, so for the active disease, one needs treatment, okay? And how this disease is detected? We detect, usually for all infectious diseases, we detect the pathogen. If the pathogen is present, we say the person is infected with this disease. But do you know a stage called latent tuberculosis? Students? Okay, so latent tuberculosis or latent phase of infection exists in many infectious disease where the pathogen is present in the individual, but you do not see any symptom. It's very difficult to detect the latent condition of any infectious disease, particularly because the pathogen hides itself inside the host, and that's its survival strategy. So to detect latent tuberculosis is very difficult, and currently no such very good detection test is available except few, but these are also not the best. And there also can be a person who is exposed to the pathogen, but he may not show up any symptom. So exposure to the pathogen, any kind of pathogen, doesn't matter whether it's mycobacterium or anything, doesn't matter, everybody will get the disease. So if you look into this slide, it's estimated that in a country like India, where the tuberculosis burden is high, all of us are exposed at certain point of time in our life to the pathogen. But about 70% of people do not harbor the pathogen, they can clear the infection very easily. So you need not to be scared if you are exposed to any infectious disease, because your immunity might be strong enough and you may not get it. Only 30% of people might harbor the infection, the pathogen, of which again 5 to 10% people will only get the disease. But rest of 90% of this 30% people do not show the symptom, but they carry the pathogen. So these are actually the individuals whom we can call that they are latently infected. But one problem with this latent infection is that they act as a reservoir of the disease, but they do not show any clinical symptom. And around the world, it is estimated that 1/3 of the world population is infected with mycobacterium in latent condition, so they do not show any symptoms. Now, unfavorable condition of the host, if immunity is suppressed by some other disease or drugs or by some other kind of physiological state, a certain percentage of these latently infected individuals can show up the disease, or the mycobacterium can get reactivated in them. Okay, so from the latent condition, they can come back to the reactivation stage. So one question obviously comes to our mind. Is why there is difference? Like when a group of people are exposed to a pathogen, why somebody is protected? Why somebody is infected, showing up the full-blown disease, and why somebody is latently infected? So here comes the question of individual variation or difference between the individual that can have some contribution towards this different outcome of exposure. So if you think of a disease spectrum, at one end we call the disease to be genetic, where the genome of the individual, if that has any mutation, even if one mutation, that is sufficient to express the disease. For example, like hemophilia or thalassemia, you know that there is mutation in the beta globin gene. So even one mutation can cause the disease. Okay, so there are a number of disease like that, which we call like Mendelian disease, and one mutation is sufficient to cause the disease. There are some another group of disease where we do not find such mutation, but multiple genes are involved in expressing those kind of disease. Along with that, there are important role of the environmental factors as well. So these group of disease are called the complex disease or the chronic disease. Okay, so here the Mendelian diseases are rare, but the effect of mutation is such that one mutation can cause the disease. Whereas in the common disease, which is called a complex disease, where multifactorial, commonly seen in multiple people, there in a population, if you screen, you will see multiple people are affected with that disease, and there is lot of effect of the environmental factors. For example, diabetes, heart disease, and so on, they will fall under this group. And it was thought that the pathogen is sufficient and necessary to cause the infectious disease. But there are a lot of evidence have come up, and now it is unequivocally proved that for infectious disease to happen, we definitely need the pathogen. But along with the pathogen, the host genomics or the host genome also plays a very important role. So here, it's no longer at the other end, but it has shifted towards the middle of this disease spectrum. And if we enlarge this portion, which I will show here, if you think of a disease triangle with the three vertices with having host at one, the pathogen and the other one, and the environment is the other one. So in case of infectious disease, the host-pathogen interaction is a very important aspect of the disease. Along with the pathogen, it can interact with other microbes. For example, if somebody is HIV infected, there are high chance that they get also infected with the MT. So here the pathogen not only interacts with the host, but it can interact with other microbes as well. And obviously, environment also has an important role. The environmental factors, I would say, you know that the nutritional status, the health status, etc., are driven by what kind of food you are eating, in what environment you are staying. So those things also play very important role. So any disease to precipitate, in case of other disease like complex or chronic disease, I would say the environment and the host, the genome plays an important role. And in case of infectious disease, the two genome, the genome of the pathogen, the genome of the host, along with the environment plays a very important role. So this complex interaction actually helps to understand the disease, making it further complex. So in infectious disease, why do we need to study the genetics? Because infectious disease is caused by infection. So there are a lot of evidences coming up and has established that yes, in infectious disease also it's necessary to understand the host genetics, because the genetics determines the susceptibility of that individual towards not only the pathogen but also towards his or her response to treatment as well. There are evidence that this infectious disease also has a familial aggregation, meaning in a family, many people are infected with the disease. You can argue that yes, they are staying close, they are in the same house, so there are high chance that the pathogen will spread from one person to other. That's true. But there have been evidences where different, person who are unrelated, meaning not of the same family, but living together. So family members who are related, genetically related, are more prone towards a disease, whereas another person sharing the same household, but from a different family background, might be protected. Okay, so there are evidence. Also, the another piece of evidence is high concordance among the monozygotic twins. So there are a lot of experiments done in monozygotic or dizygotic twins. Twin study is a very important piece of evidence for the genetic determinants or to identify the components in any kind of disease. So monozygotic twins, you know that they are 100% identical. So their susceptibility or the concordance rate of infectious disease among the monozygotic twins are higher than the dizygotic twins, whose sharing of the genome is not 100%. And also there has been an incident, an historical event called the Livet accident in 1929 that also gives us a strong evidence that yes, in infectious disease also, the genetic plays an important role. So what was that incident? In Germany, newborn babies, they were given the BCG vaccine, and in one place for Lübeck, this incident happened. So about 251 children babies were inoculated with BCG vaccine they were given, and after certain time, few months, it was found that the babies show the symptom of tuberculosis, and out of 251, I think 77 infants died. Okay, so it was a disaster, and then on investigation, it was found that the BCG vaccine that was given had some problem. So initially they thought that the vaccine has been, the pathogen in the BCG has been reactivated and that caused the infection. But later on it was found that that is not the case. Actually the vaccine was contaminated with Mycobacterium tuberculosis. So actually along with the vaccine, the babies had received the live Mycobacterium tuberculosis. Now the fate was that out of 251, 77 infants died. Many of them, about 39 of them did not show any symptom of disease, and some of them showed mild symptoms. So again we see that a few slides back that I have shown you that there is differential expression or differential response towards the infection. So this study, even though it's an accident, this again proves that the individuals, the inter-individual variability among different individuals actually determine their response to the disease. So it clearly demonstrates that study of host genetics is necessary. Now, how would one go to study the host genetics or the determinants of host genetics in case of tuberculosis, in case of infectious disease? So there are multiple ways to study. I will just mention some of these and then show a small part of data that we have generated. So one could be a family study, family-based study. You compare the genome of, in a family, multiple members of a family, where someone is infected and someone is not infected. So you can estimate or you can guess that people who are infected, they lack some immunity or some factors, whereas people who are protected, who do not have the disease, have something strong in them. So you compare the genome of both of these groups and you come to a conclusion or find the difference between them, which can give you an idea like what are the genetic factors that's playing part in those infected individuals. The other type of study is unrelated case control. Here you are not taking family members, you are taking just unrelated individuals. Suppose I want to carry out that study in a hospital or even people here, I just randomly choose people who are patient, who are infected versus another group who are disease free. And again compare their genome and see that, and after that you have to obviously do some statistical calculation to arrive to a meaningful conclusion that what are the factors that are present in the infected individuals versus what is not present in the uninfected individuals. So this is just a diagram to show what exactly I mean. So in a family, you compare the filled boxes are the individuals, males and females, and in the association or the unrelated set of study, you take just randomly screen people from a community, from a hospital, from a population, etc. So these are the two basics or two kind of studies that one can conduct to find the genetic contribution of our genome in any disease. Okay. So carrying out the first type of study from like more than 20 or 25 years, in case of tuberculosis, people have identified some in different chromosomes that might harbor certain genes which has contribution towards determining susceptibility to infection. Okay. So I have given some examples here. I'm not going into the details. One interesting feature I would mention here is a study here done on South African population, which was, so another feature of this population-based study is that when you do it in one kind of population, your conclusion has to be validated in a different population or a different set of samples. Then only you can say, okay, your finding is true. So this study was first carried out in South African population and later validated in European population as well. And they found that two regions in chromosome 5 and 11, they determine the tuberculin skin test sensitivity and positivity. Okay, so you know what is tuberculin skin test. If somebody has some symptoms or the physician suspect, they do that two tests whether we are infected with the mycobacterium. But since we receive BCG, in many cases it's not possible to conclude. But anyway, so in European population or in USA, BCG is not given, so they are sensitive to the test later on, not like us. So this two locus, actually later on it was verified in other population, shows some indication that some genes are present in this region, which determines the susceptibility or sensitivity towards this tuberculin skin test. And later on it was found that TNF-alpha is a cytokine you know, which is secreted or overexpressed in any kind of infection. The TNF-alpha actually lies in the 11B region. So this way, doing the linkage study also, in linkage study or the family-based study, what we are looking? We are looking whether the disease gene is in all individuals, some common region is segregating among the infected individuals or not. So the major interest is those common regions and to find a gene in that region. So that way we can identify genes which could have role in this infection. Now later on it was found that in the chromosome 5 region, a protein called SLC6A3, which is actually a transporter. This gene resides there, and later on, even though it's a neurotransmitter transporter, which apparently one could think that this has no role with infection, but it was found that yes, it also has role in infection. It was demonstrated from SLC6A3 knockout mice because it was reduced delayed type of hypersensitivity as well as reaction to certain antigens. So this also helps to identify genes whose function we know in one area of physiology, but it could have diverse function and it could also have role in other aspects of the cellular physiology. Okay, so the unrelated case control, in other words, is also called association study, where we target certain genes with some prior knowledge, and this is called candidate gene approach. Why this is called candidate gene approach? Because based on certain prior knowledge, we are considering that okay, this gene might have some role in the infection. So I'll give you some examples. Say in case of infection, many genes like chemokines, cytokines, they have very important role. So these are nothing but small peptides and proteins. So the genes coding for cytokines and chemokines could be a candidate gene. Okay. Then there might be other genes which are playing role for bacteria or virus killing. Some transporters which are transporting certain metal ions across the membrane, and these transporters are also very important. Then cell death is a part of innate immune response towards many pathogens. So genes that control or play part in cellular apoptosis or other kind of cell deaths also could be a possible candidate gene. So this way one can think about to pick and choose candidate genes. Okay. So, how these are, what assay is done on these genes? Let me show you. Actually, to know, we are considering what are the variations present in the genome. So, these, if you like consider these three individuals, or if we analyze the genome of people present in this hall, you will find that in certain positions they differ. And this difference in the nucleotide is called the single nucleotide polymorphism. Why it's polymorphism? Because it is present in the general population and it's not a mutation. For mutation, the frequency is much less, but the effect is much higher. But in polymorphism, it is present in higher percentage. It's present in the normal population with little effect. Okay, so if we compare these differences in individuals or in two group of people, diseased and normal, we can, in certain cases, we can assign or we can try to find out what these changes or what these polymorphisms are biologically doing to precipitate the disease. So this is one way what we consider in case of association study or the case control study. So we assess the polymorphisms in different individuals, and that way multiple genes in case of tuberculosis were identified, one of which is NRAMP. So this is actually a solute carrier protein, and it was found in multiple studies done with tuberculosis case and controls. Later on it was found that it also has role in macrophage function. And macrophage is is the first cell or the major cell where the mycobacterium infects and resides inside that cell. The other piece of evidence, see what was found in with linkage analysis, the region 50. So this also harbors nearby the NRMP gene, and this gene is not only implicated in tuberculosis, but some other infectious or autoimmune disease like sarcoidosis, where certain features are similar to tuberculosis. Not exactly, but certain features. So this way, one can identify the host genes which are implicated in determining susceptibility towards the disease. So here is a long list of genes. I am not going through them, and many more are known. Now, after the era of these individual genes came the era of genome-wide approach. Because in this candidate gene approach, you are biased. You are having a prior knowledge. So you may not discover new things there. You can establish the earlier findings, but you may not get the new things. Later came the genome-wide studies where you are totally unbiased. You are looking into the whole genome. Okay. So whole genome studies have been also performed in case of tuberculosis. The list is long, the whole list is not here. You can see that this has been performed in different parts of the world. Okay, so these studies are required in multiple populations because finding in one population may not be validated in the other, as we know that the population might differ in certain percentage in their genome. Okay, so in the genome-wide studies, one has to validate the finding in the other population from other countries also. So what are the challenges here? The challenges are you identify multiple polymorphisms, but it's very difficult to find what is exactly the causal variant. Out of that millions of variations, out of that millions of polymorphisms, which is actually the problem? Okay, and sometimes the population inadequate sample size, you need thousands and thousands of samples in order to attain a statistical significance. So inadequate of sample size or population specific variations might be there. Then if your phenotyping or study design is inadequate, then also you might land up with a wrong result. And two more things in case of infectious disease is very difficult or challenging to determine. That's the degree of exposure. Like when you are exposed, it is very difficult to understand. That brings a degree of complexity. And also, what are the different strains? Some strains are more virulent, that can cause more havoc than the non-virulent or moderately virulent strain. So these are the few challenges, even though a lot of discoveries have been made towards the contribution of host genome, but there are still a lot of challenges. So in host pathogen interplay, so these are some of the important factors that we will look into. From the host side, the response of the host towards disinfection, the underlying genetic variations towards those variations, and in the pathogen, the strain, the altered gene expression of the pathogen, because the pathogen is also in the hostile environment inside the host. So in order to establish or in order to survive, the pathogen also has to fight with the host immune system. And so this balance and the fight between these two determines like who will win. So if the host wins, it can eradicate or it can clear up the infection. If the pathogen wins, it is trying to establish itself. So we, in our research, we mostly focused on the cytokine response and the underlying genetic variations. So to understand this, we adopted a case control study. So what are these cases are the tuberculosis patients who are clinically confirmed by the physician through pathological test and so on. And we took the controls as their household controls, who are staying in the same household, sharing the same environment, because our aim is to nullify the effect of environment. If environment and genetics both change, it is very difficult to find what is the contribution of each factor. So we took person from the same household who is not infected. And what were our selection criteria? Our selection criteria was that the patient has to be clinically symptomatic and culture positive, meaning we have confirmed the presence of pathogen. They have not received any treatment. That means first time they have the disease and they are coming to the clinic. So if they start receiving the treatment, the symptoms will subside, the immune response will change. So we do not want to include those kind of patients. Obviously, HIV is a major factor of co-infected factor in case of tuberculosis. Again, to keep the situation simple, we did not include any HIV-infected patients here. And these patients are first time having the disease, they did not have any past history. And what did we consider for the household contacts? So household contacts are sharing the same household with them. In spite of long stay, they are still free of disease. Clinically asymptomatic, we tested them for the presence of pathogen and they are all negative. They also do not have any past history and of course they are also HIV. And specifically, actually we consider the spouse because in a household, the spouse, they are sharing the same environment, yet they are from different genetic background. Assuming that they are from different genetic background, we consider the spouse because that will be ideal to carry out the genetic study. So what did we do? We collected some amount of blood, isolated DNA, and I'm not going to the details. This is a technique by which, after isolation of DNA, we have assayed the variations or the polymorphisms that are present in the genome of the uninfected person and the infected person. So this is actually a multi-scale study. It is not that we looked into one position or one polymorphism. We looked into 2.5 million variations in the genome. So what was the result? We arrived ultimately to this many samples. 230 participants were there, 119 pairs, 119 cases, and 119 controls of the household contacts. We had analyzed these many markers. So here marker means, I mean that the polymorphic or the single nucleotide polymorphic position in the genome. Okay. So after assaying that polymorphic, what is the status of those markers in these individuals and performing certain quality controls, we arrived that 29,000 markers would be informative. Okay, so you start down to very low frequency, but the chances are that after all kind of quality controls and so on, even though this is less compared to your initial starting point, but this could be informative. So you shed off the unwanted or the junks in between throughout your analysis. And with these markers, when we perform certain analysis, which I am not going to discuss with you in details, that will take a long time, what we found that, so this plot is called the Manhattan plot, because it looks like high-rise building, and in this axis, we are plotting the data obtained from different chromosomes, and this axis, we are plotting something called log p value, which actually gives you a sense of significance. Okay. So more it is negative, more it is significant. So we found that two such nucleotide polymorphisms, which the nomenclature is like this, RS and with then some number, one present in the SLEC15 gene and two present in the HLA-DRA gene, we could identify that is specifically present in the individual who are infected with the disease. Okay. So we looked into what are the function of this gene. HLA, you know, it has well documented role in immunity and towards adaptive immunity as well. And CDX is a lectin, which lectin specific receptor which is present on the immune cell of human or host, which actually detects the sialic acid present on the pathogen. So it acts as a receptor. So these two polymorphic sites on these two genes we found. There were certain other polymorphic sites that we had also identified, and we considered the top few to. So once you discover the variation in the genome, that doesn't end your study. So now you have to establish what biological function actually that variation is doing. So unless you prove that biologically, it doesn't have any meaning. Okay, so what could be the possible implication of those variations? There are certain evidence and there are certain predictions. There are use of lot of bioinformatics to predict probable function of these variations, and later they are experimentally validated. So this is the strategy when you work with the genetics, that you identify the variations, you predict certain function, and then you have to experimentally validate them. So if a variation is present in the coding region and changing the amino acid, you are in a good position, your life is easy. But it could be also a synonymous change. It doesn't change the amino acid. Okay, so what it can do? Prediction is that it can change the expression level of the protein. But these are predictions, one has to experimentally validate. But lots of variants are found in the non-coding region. So even genome has lot of non-coding region. Non-coding region also has some important role. It is not that the non-coding region has no function. So in non-coding region, it is promoter. One can again think of that the promoter change of these nucleotides might become active or less active and alter the expression. If it is present in the splice site, it can alter the splicing of the protein, making a different protein completely non-functional or hyperactive protein. If it is present in the UTI, it might be associated with the stability of the protein, or it might also interact with the microRNA, and that such interaction can alter the function of the protein. If it is present in the introns, that can also lead to certain other features. EQTL, meaning expression quantitative trait locus, which actually express or controls the expression of other proteins. Okay, so even polymorphism or variation is present in a non-coding region, it can regulate expression of a different gene, or it can regulate the expression of the same gene. It can act as an enhancer for other genes. So non-coding regions are not that they are junk. They have lot of regulatory functions. So the siglet or the HLS names that we identified to be associated with tuberculosis, we find that these EQTL, these expression EQTLs are tissue specific. We know that not that all proteins are equally expressed in all tissues. They have some tissue specific expression also. So these HLA SNPs or the polymorphisms that we identified, they are EQTL in lung for two other HLA genes, and in the whole blood they are EQTL another HLA gene.
লাস্ট কথা তো
হ্যাঁ তারপরে
Blood is a very
প্রেজেন্টেশন
relevant
একটা
for
সময়
tuberculosis
প্রেজেন্টেশন
because
বলে যে
this
একটা যদি বেজে যায়
is a
প্রেজেন্টেশন
case of
খাওয়ার পরে
pulmonary disease and also the immune cells present in blood,
প্রেজেন্টেশন লাঞ্চের পরে
systemic,
হবে
the flow of blood also has lot of immune response, right? So the
আমি কি জানি
lung and
আমাদের তো
blood
নেই
is very
ক্যাস্ট্রল
appropriate tissue to consider the changes in tuberculosis. This that way we think that our finding has some true
সব
biological
একসাথে
value.
ঢুকে দেবে
Besides that, the cyclic gene, the variation that we found in cyclic gene had a a synonymous change. So that change was in the nucleotide, but it was not changing the amino acid. So we were puzzled and thinking what could be the functional implication of such change. At the same time, when we published our paper, just a few days back, another paper came out where they found that another polymorphism in the same gene, the cyclic gene, cyclic 15 gene, was associated with the infection of candidiasis. Okay, so next, therefore it shows that our finding was true and this cyclic gene is of course associated with multiple kind of infection. And then we tested their snake in our samples and we found that yes, that snake which was only 1 kb apart from our polymorphic size that we found is also associated with tuberculosis in our samples also. So this suggests that even though the change that we found was a synonymous change, but this snake could act through another snake, which is like 1 kb apart. So this way, once one finds the genetic variations, it's extremely important to find the functional implication of such variations. So to summarize, what we found is that in the HLA gene, two novel variations we could find which could act as an equivalent for certain gene expressions, other HLA gene expression in lung and blood, which are very relevant issues for the tuberculosis disease. And also, even though this snake doesn't change the amino acid, but it changes the amino acid and known to modulate immune function, immune alteration. So the cyclic genes might have this altered function and act to the other polymorphic site, determining the susceptibility towards infection and immunity. So currently, actually, we are working more basically onto the function of this. I will just finish going through a few slides, talking more, just a glimpse of what we do in our lab. Like we have taken multiple actions to understand the genomics of tuberculosis infection. So as I said, that the whole environment interaction is important. We are focusing on that, looking into population-based studies. What next? What are the susceptible factors? We are looking into the expression profile of the cases. Also, we are looking into whether we can identify any biomarker for the active state of disease. And obviously, all these genomic findings we are validating in the cell-based models for the functional importance. So these are some specific areas where we are taking multiple approach in a case control study. We are trying to establish the role of this hemotype that we identified as an active marker of disease. What is its biological function? What downstream biological function does it perform? And also we are trying to identify more host novel factors using genome-wide CRISPR screening. Because see, for tuberculosis, there is only one vaccine and few drugs are used for long time. So there is no progress in vaccine development or newer drugs, and obviously all the newer drugs are very, very toxic. So in last few years, host-directed therapy is an avenue which is getting importance, and so for knowing or performing host-directed therapies, you need to have more knowledge on what are the host factors involved. So in GWAS, you get an idea about the gene. CRISPR screening also you get to identify new factors with functional application. So this is what is ongoing, and one or two slides about my institute. I would say particularly this is for the students. So what do we do at NIBMG? our motto is excellent in genomics for health. And we have 4 groups or 4 petals of the flower. One group working on the chronic disease, another group working on infectious disease, a group on cancer, and we have a group of statistical genomics who help us to analyze the big data that we generate from all these genomic, transcriptomics, so on the big large-scale multiomics studies. They help us, they develop methods, they help us to analyze the data to a meaningful conclusion. Okay, so our aim is to understand a disease at the genome level, at the transcriptome level, any alterations in the epigenetic and functional protein. So, in few words, predict, prevent, and cure. Okay, I will also, this is for the students, here are pictures of the facilities that we have. And also a glimpse of the campus life. Ours is a residential campus. You have to, if somebody joins there, you have to stay there. And the different programs for the students that we have. We have integrated MSc program, PhD program. Also, we take students for short-term training. And if you keep your eye on our website, throughout the year, we have a lot of training and workshops. So, if you are interested, you can check on our website. With this, I come to the end. I would like to thank the patients and their family members who has given us the samples. For them only we could perform this study. The students who were involved in this study, and Professor Bhaskar and Dr. Shomshiti Bhattacharya, who helped me with this analysis, the strong team on the study. I received the funding from Department of Biotechnology and also the institutional funding, which helped me to carry out all this work. And I will end here. And thanks to all.
Thank you, ma'am, for such an engaging talk. I would request Dr. Tilakraj Maiti sir, Assistant Professor, Department of Biotechnology, Haldia Institute of Technology, to kindly come forward and felicitate our distinguished speaker with a token of appreciation. Ma'am, can you please?
তারপর সিনিয়রদের হবে তারপর জুনিয়র
I would now request Mrs. Sonali Mithya ma'am to please come forward and felicitate our co-chairperson, Professor Shubhrama Gupta ma'am, with a token of gratitude. Now we will be moving on to the poster presentation of the first session. So the first group is Shobhata Panigrahi, Tuhin Ponda, Sunit Maiti, and Avik Shamunto. And the topic of presentation is Survey of Cryptograms of Darjeeling District for their current status and search for a solution. And they are coming from the Department of Botany, Prabhat Kumar College. Contact. And the presenters for this poster will be Shobhata Panigrahi and Sunit Maiti.
রোহিত কোথায়? রোহিত কোথায়?
Good afternoon to all of you present in the seminar hall. Here we means myself, Shobhata Gray, and my friend Sunit Maiki are from Rathmar College, Ponte. Today we are here to share a poster presentation based on survey of cryptograms of Darjeeling district for their current status and search for a solution. At first, we have to know what are the cryptograms. Crypto means hidden, and gamy refers to the reproduction. So these plants, those plants which are unable to produce flowers and fruits, instead they reproduce with spores, are called cryptograms. So few days ago, we went on a educational trip to Darjeeling district. There we found such cryptograms. We found that they are in a serious threats. That so we did a survey report and made some solutions on them to preserve them in XE2 system. For such evidences, we took some details pictures which are also shown in our poster.
এন্ড কখন হবে?
দেরি আছে এই দুটো পোস্টার প্রেজেন্ট হবে তারপরে problems are the cryptograms only available only in the hilly region of northern part of West Bengal like Darjeeling, Mirik, Kalimpong, Karsian, etc. area. But nowadays, some major problems are noticed, such as soil degradation, urbanization, education, environmental pollution, and climate changes are causes serious threats to their existence. So we need to special care for these lower class plants for to preserve it in exit condition. So next part we will discuss my friend Sunit.
So to come out from this problem, we proposed a system. In this system, in which different hilly-like environment are created in the system. This system, different types of endemic plants or species are grown in the system. This system required less investment on monitoring. Also, different types of carnivorous plants are grown in this system in non-tropical and less humid region. So students can study on these plants. To make this system, it required less area and it is also cost effective. The system is maintained free of cost and run by solar energy so no extra electricity are needed. The water that used in the system, that is mainly rainwater, we collect this water in rainy season and used in the system. So we can reduce the water waste. Moreover, biological curiosity can be resolved out for the students. So next is Subodha.
So, these lower class plants are very much essential for medical purposes like drug industries and also for our botanical laboratory work. By the above proposal, we can preserve these plants in a cost-effective way and prevent them from endangered species. Moreover, it can be provided in rural or semi-urban area where still electricity are not sufficient or infrequent. So there, by the use of solar energy, we can run this system. Our future plan is to develop the prototype of this system, and finally our ultimate goal is to provide a system for common researchers, universities and colleges for further research work. So finally, a big thank you for everyone for giving us this opportunity to present our topic. Thank you.
Do we have any questions from the audience? Okay, then thank you for the presentation. Up next, we have Shashwati Gayan and Dr. Shomitra Kaur, who will be presenting on turning the morphology of the zinc oxide nanoparticles by the solvothermal technique. And they're coming from the School of Applied Science and Humanities, Haldia Institute of Technology. Participants should also know that the time limit is 10 minutes. It is 7 minutes for presentation and 3 minutes for the question answer session. Good afternoon to all. I am Shashwati Rai from Department of Applied Science and Humanities, Haldia Institute of Technology, Haldia. Today, I am going to present my poster on tuning the morphologies on zinc oxide nanostructures which can be thermal process. Zinc oxide is a semiconductor with wide bandgap energy 3.3 electron volt. Also exhibits a large exciton binding energy 60 MeV at room temperature. Zinc oxide is a room 6 semiconductor having potentials in various optoelectrical bioimaging applications. It has also applications in flat panel displays, sensors, light emitting diodes, laser diodes in ultraviolet visible range. 1D nanoforms of zinc oxide is a highly suitable candidate for nanoscale applications like room temperature laser, field effect transistor, nano resonator, etc. In our lab, we synthesized one-dimensional zinc oxide nanostructures by using solvothermal process. In this process,
chemical reaction takes place in a solvent at temperature higher than the boiling point of the solvent used in a closed vessel. Also, it involves high pressure. It is a simple process to synthesize any nanomaterials. Also, it is not expensive. Some basic features of this process are almost all materials can dissolve in any solvent at high temperature and pressure. Solvothermal process is characterized in mild temperature condition. High pressure also important for enhancing the rate of chemical reaction. Solvent is the most important influencing agent to determine the morphology of the products. In our laboratory, first we have synthesized 1D nanostructure of zinc oxide with water-based ethylene diamine solvent on metallic zinc foil. Here, the same image are shown. Same images are shown here. First two images prepared only for water. Here, ethylene diamine not used. Then we take different volumetric ratios of ethylene diamine and water ratio, and we get second and third image. Last one is the graph last one is the graph when we use ethylene diamine only. This process, the rate of this process was very high, so the structure we get was in micrometer range, not in nanometer range. Uh, here the reactions, ethylene diamine and water, we get OH minus ion, hydroxide ion. During this process, hydroxide ion and zinc ion reacts and zinc hydroxide form. At high temperature and pressure, zinc hydroxide dissolves again and form zinc ion and hydroxide ion. When the temperature again increases, the process continues and reach a saturation state. Zinc ion and OH minus ion combines, reacts, and zinc oxide forms. To make the reaction mechanism slower and to get structure in nano size, we get ethylene instead of water with a basic environment on metallic zinc foil. First two same images of the product ZNO synthesized at 170 degrees centigrade and 200 degrees centigrade. In this case, no basic reagent was present here, not any OH was not present. In the group of second image, moderate amount of NaOH was used. Here, zinc nanorods and nanoscales were formed. The last same image of the groups. These state ZNO nanorods. Third group of image showing TEM and HR10 nanopins and nanorods exhibiting solid structure of them. If we when we made the solution strong basic with NaOH, we got first zinc oxide nanotube structure, and finally we got paint brush structure of ZNO. In the right hand side, a schematic representation of growth mechanism for ZNO nanostructure are shown here. The produced nanostructure was in hexagonal structure, and Miller indices for corresponding image are shown here. Structurally, the wood-type structure zinc oxide crystal is described as number of alternating planes composed of four-fold tetrahedrally coordinated oxygen minus ion and zinc Zn2 plus ions stacked alternatively along the C-axis. The produced zinc oxide nanoforms result in a normal dipole moment and spontaneous polarization along the C-axis having Zn2 plus ion and O2 minus ion in another side. Zn2 plus in the positive side and O2 minus in the negative side. Here, one face of the hexagonal sheet is zinc rich from 0001 plane and oxygen minus ion negative rich. It forms 0001 bar planes. Uh, 0001 bar planes.
আর হবে না মানে সেকেন্ডটা তো
These Zn2 plus ion and O2 minus ion, Zn2 plus positive ion and negative ion O2 minus combines, reacts and the O2 minus rich negative surface can attract new ZNO structure or the opposite species of opposite surface. Growth rate was maximum at 001 direction. That's how we tune the morphologies of zinc oxide nanoparticles by survothermal technique. That's all about my poster presentation. I would like to thank my guide, Dr. Soumitra Kaur, of the Institute of Technology, Haldia. Thank you.
আমি পাশে তো দিতাম কেন কাজ চলছিল সেকেন্ড ওই তো প্যাক করা উনি বড় লেভেল
Do we have any questions from the audience? Thank you, Swati Nair, for the presentation.
Okay.
I would request our